Welcome to my video for the Student Research Video Competition at Cereals and Grains 20. My name is Ana Magallanes Lopez. I am a PhD candidate in the Cereal Science program at North Dakota State University, advised by Dr. Shenai Shimshek. My research is entitled Ingredients Extracted from Dry Beans, Chemical, Structural, and Functional Characterization. So let's start from the beginning. As expected from my Latino roots, I love beans. Fortunately for me, I am in the #1 state of dry bean production, North Dakota. But hey, there's not only one type of bean. Actually, in the US, several dry bean market classes are grown, such as pinto and black beans, among many others. But why caring about beans? Well, these little pulses are an important source of proteins, starch, minerals, phenolic compounds, and dietary fiber. So many marvelous things perfectly packed. However, my research is centered in two of these ingredients, phenolic compounds and dietary fiber. Now, phenolic compounds are secondary metabolites with potential health benefits due to their antioxidant properties. On the other hand, dietary fiber is a complex mixture of polysaccharides that resist enzymatic digestion in the small intestine and can undergo a complete or partial fermentation in the large intestine, thus promoting beneficial physiological effects in human health, like anti-inflammatory activity. So, with this, I want to test the pro or anti-inflammatory effects of the soluble dietary fiber extracted from the dry beans on murine cells, and also work with adipose tissue cells to evaluate if the extracted soluble dietary fiber can modulate the production of some signaling molecules associated with obese-related chronic inflammation. Therefore, this research will enhance the human health while promoting the market value of the dry beans. In this sense, my research falls in the processing food system category because of the extraction of potential food ingredients. As in any chain, the links are interconnected, so my research also impacts the pre-production category since phenolic and dietary fiber contents can serve as a criterion for selecting varieties that will be used for the extraction of these ingredients. Furthermore, if the ingredients are successfully extracted and characterized, the production category will also be impacted as nutraceutical products might be developed. Therefore, my research directly contributes to the goal #3 of promoting good health and well-being from the FAO Sustainable Development Goals. But now, how am I achieving these? Well, 2 dry bean market classes grown at 3 different locations in North Dakota were used for the extraction of free and bound phenolic compounds that then were analyzed. Then I utilized an enzymatic gravimetric method for assessing the dietary fiber content of the samples. I move on with the soluble dietary fiber extraction of raw and cooked flour samples. Then I characterize the functional properties with rheological tests. I move on into the structural characterization with HPLC, YEC, and ion NMR technology, which I am still working at. Now, in the second phase of my project, I will use cell culture experiments to assess the immunomodulatory properties. Now, so far, the growing location has denoted differences on the antioxidant potential of both pinto and black beans. The extracted soluble dietary fiber is mainly composed by arabinose, xylose, galactose, and in lesser amounts, mannose and glucose. I am expecting to see structural correlation from the soluble dietary fiber with the anti-inflammatory responses, in addition to an impact in the production of these pro-inflammatory signaling molecules of the adipose tissue, also known as adipokines. Therefore, the soluble dietary fiber is expected to serve as a functional ingredient for wheat-based products, alleviating the obese-related chronic inflammation. With all this, the results from my research will be significant to market the immunity enhancement of dry bean consumers. Thank you.